Hello everyone, it is my great honor to share with you our study entitled Soil pH Modulates Microbial Nitrogen Allocation in Soil via Compositional and Metabolic Shifts Across Forests in Japan. Ammonium release and uptake by soil microbes are key components of forest nitrogen cycling, controlling nitrogen fluxes that affect plant productivity and ecosystem nitrogen retention. However, because these processes involve diverse pathways across many taxa, their community-level regulation remains poorly understood. Here, we combined rRNA gene sequencing, shot genomics, and 15 nitrogen tracer assays to examine how microbial community regulate nitrogen cycling across forest ecosystems. Pedogenic factors, climate, soil type, and vegetation, deterministically shaped soil properties, carbon, nitrogen, and pH. Vegetation, together with precipitation and temperature, primarily controlled soil pH, which was higher in coniferous than in broadleafed forests. In contrast, soil type, modulated by temperature and precipitation, governed soil carbon and nitrogen contents, with andesols showing the highest and regosols and acrosols the lowest values. Soil properties had stronger impacts on microbial communities than pedogenic factors, with bacterial communities showing greater explanatory power than fungal ones. Soil nitrogen content dominated microbial abundance, additionally influenced by mean annual temperature for bacteria and by the C/N ratio for fungi. Both increased with higher nitrogen content. Revealed that soil pH was the major driver of bacterial and fungal community composition, explaining 61.72% and 38.01% of the variation, respectively. Microbial taxonomic composition was therefore highly correlated with soil pH. Partial least squares path modeling revealed that nitrogen cycling rates, ammonification, nitrification, and ammonium/nitrate consumption increased with bacterial abundance and were also influenced by bacterial and fungal compositions. Nitrogen allocation, reflected by ratios of ammonification to ammoniumfication and nitrification to ammonification, was mainly governed by bacterial composition. High pH soils exhibited stronger nitrogen release and nitrification. Microbial richness was the key determinant of nitrogen cycling gene abundance and nitrogen allocation. We then examined how the relationship between microbial community composition and nitrogen cycling varied with environmental conditions. In low pH broadleaf forests, nitrogen cycling rates were driven only by bacterial abundance. In contrast, in coniferous forests with near-neutral pH, community composition contributed to nitrogen cycling rates to a similar extent as abundance played a dominant role in nitrogen allocation. In andesol forests with higher carbon and nitrogen contents and greater bacterial richness, nitrogen cycling rates were controlled by bacterial abundance, while community composition primarily determined nitrogen allocation. In cambisol forests with lower carbon and nitrogen contents and lower bacterial abundance, community composition exerted stronger on both nitrogen cycling rates and nitrogen allocation. To assess whether context-dependent effects of community composition on nitrogen cycling were mediated by metabolic capacity, we characterized microbial metabolic potential using the composition and abundance of KEGG metabolic modules. Community composition strongly correlated with metabolic capacity and soil pH. Nitrogen metabolism, nutrient transport, and two-component regulatory modules related to heterotrophic nitrate reduction, denitrification, and nitrification all increased with pH. Likewise, phosphate and amino acid transport modules, and most mineral and organic ion transport modules, as well as respiration regulatory functions, were positively correlated with pH, while stress response functions decreased with pH. Microbial control of ammonium allocation, between retention in biomass and transformation to nitrate, is jointly governed by community composition and metabolic capacity. Pedogenic factors shape soil pH and organic matter, which determine microbial structure and regulate nitrogen cycling and allocation. Microbial abundance primarily affects nitrogen cycling rates, while composition and richness influence ammonium release or retention through metabolic constraints. Among soil properties, pH acts as a key regulator. Acidic soils favor stress-tolerant microbes that retain ammonium, whereas near-neutral soils support energy-efficient taxa enhancing ammonium release and nitrification. Overall, microbial composition and metabolic capacity jointly regulate nitrogen emission, with soil pH serving as the central environmental filter linking soil properties to ecosystem nitrogen cycling.